the title of my presentation is Gene Network Inference by Fusing Data from Diverse Distributions. So the most straightforward approach to network inference is a similarity-based approach, where we assume that two genes, if they are functionally related, they share high similarity with respect to a particular data set. Which means that given a data set, we measure somehow the similarity between a particular between two genes, and if this similarity is above certain threshold, then we link these two genes. So in contrast, there is a model-based inference, which rather focus on local dependencies between genes and assumes that the behavior of a particular gene is affected by behavior of a small number of genes. So in model-based approaches, the task is actually to identify which are those small set of genes that can predict well the behavior of a particular gene, and this defines the neighborhood of a particular gene. Okay, so what we focus here is on the model-based approach. But before that, most standard techniques that are based on models assume that data is Gaussian distributed. Okay, so this is a nice assumption, but in many data that's being generated recently, such as data from RNA sequencing, we can see that these actually aren't Gaussian distributed data. So for example, RNA sequencing, what we have is the counts of transcripts that map to specific genomic location. So this could be better modeled with Poisson distribution or negative binomial distribution. Similarly, for example, we can have mutation data where we have information about various types of mutations or copy number variation data, and these are categorical data. And again, this type of information could be better modeled with Dirichlet distribution or multinomial distribution. So it would be nice to have a method that would be able to handle diverse set of data distributions, but not only that, at the same time, this method would be nice to have a method that could treat various data sets at the same time, although these data sets could arise from non-identical data distributions. So this is the main focus of our work. And for that, what we use is undirected graphical models or Markov networks. So here, let's assume we have a random vector. So this random vector can be seen as a list of random variables, and each variable is a gene. So then an undirected graphical model is defined as any distribution that is defined on this set of random variables and that satisfies certain Markov independence assumptions that are encoded by the network that encodes different dependencies that can exist between the genes. So this network is what we would like to infer from the observations that are given. And the nice result about undirected graphical models is that it's known that each such distribution can actually be written in a such form. It can be written as an exponential of a weighted sum, where this weighted sum goes over all maximal clicks in our network. So you can see that in this is our case. Here we focus on a simple case of pairwise Markov network. So we have the terms that correspond to nodes and the terms that correspond to edges. Just recently, last year, it was shown that if we make some further assumptions about what the conditional distributions of individual nodes look like, we can make some further observation, and this is the one that these potential functions, so the phi terms here, can be a priori set and can account for various distributions, actually for any distribution that comes from exponential family. So an exponential family is a widely popular distribution family, which includes not only Gaussian family, Gaussian distribution, which we all like, but also Poisson, negative binomial, Bernoulli, gamma, and so on. So at the end of the day, what we would like is estimate these theta weights, the weights that corresponds to gene weights and some weights that corresponds to interactions between genes. And our task of estimating the network structure in undirected graphical model reduces to estimating these weights, whereas for the potential functions, so the phi functions, we can use the sufficient statistics of various distributions that are out there. So, okay, so this would be our first goal. So we can perhaps treat various distributions, but we want to do this simultaneously. So how we approach that? So we say, okay, so we will parameterize these theta parameters such that we will impose some latent factorial structure. And we will say that gene weights will be actually modeled by some latent low-dimensional vectors. And what these vectors encode are the membership of a particular gene to different latent components. And the gene-gene interactions will encode the interactions between two genes, their dependencies, and this will be written as a product of the latent factors that correspond to the both endpoint genes and some latent matrix which accounts for interactions between all combinations of latent components. So this is an important view which actually allows us to fuse data from different distributions. So if we have just a single data distribution, let's say we have in a single data set that follows a particular exponential family distribution, then how can we find U and V? We find U and V by solving a sparsity constraint maximum likelihood problem, and we can estimate the optimal U and V, and then based on that define the neighborhood of each node in the network. But this is not our final goal, so what we have are many data sets. So let's say we have at least one more. So we have a data set Y, which follows a different data distribution, and we can assume that it's parameterized in a different way. And this data has particular, for these parameters in Y, also have the particular latent matrix U and W, and we also have to estimate that. So the point here is that we achieve fusion by sharing some information between these related data sets or distributions that we have. And how do we share information between these data sets is that we reuse some latent factors. So in particular, there is latent factor of genes U that is shared in both data sets across both distributions. And the important part here is that a good latent factor for each gene should maximize the likelihood of both data X and both data Y. Okay. So at the end, what we solve an optimization problem that goes through various data distributions that we have, and we have some factors that are specific to each distributions. These are the W matrices that consider the interactions between latent components, and the U matrix, which allows sharing information between related data sets. So once we are able to solve the optimization problem, we want to define the neighborhood, and the overall estimate of the network is defined such that we stitch together the neighborhoods of individual nodes, and the two nodes in particular are connected in the network if their interactions are non-zero. So that means that a particular gene is predictive of the behavior of another gene. That's why we connect them. So this is aligned with the theory of Markov networks, and at the end we can have a network. We get a network like this. So let me just interpret this in the context of our neighborhood. So we can see that gene 2 and gene 3 are not connected. So, and what does this mean? This means that gene 2 and gene 3 are conditionally independent given gene 1 and 4. So intuitively, this means that if we have the measurements of the first and the fourth gene, then we can tell everything about the behavior of the third gene if we have the first and the fourth gene measurements. So overall, our approach is called FuseNet, and what it's able to do is consider various data distributions, in particular any data distribution that is in exponential family, and is able to, using the theory of Markov networks, to construct a network that can consider various data sets, but can acknowledge or can support different data distributions. So this would be the model, and the output is the Markov network that is consistent with the theory of independence and dependence between nodes that exist in the undirected graphical models. So we did various simulations and also evaluated our method on real data. So let's start with some simulations. What we did is we generated multinomial data, and in each simulation, this means that we generated a set of observations that follow multinomial distribution. So in particular, this is one variable, one gene, and its conditional distribution is multinomial. So we given this data, the goal was, can we infer the network that encodes the dependencies between nodes, and will this network be very similar to the ground truth network that was used here to generate these observations? So can we recover the multinomial network successfully? So we did this for various types of network topology, such as scale-free and small world networks, which are known to resemble certain structures that appear in biological networks. And what we measured is the proportion of edges that were present in our inferred networks and that were also present in the ground truth network, which is the true positive rate, and the higher is, the better is the algorithm. And the alternatives is the false positive rate, which is the edge that was present in the inferred network but was not there in the ground truth network. So the lower number indicates better performance. So in particular, in the scale-free network, we see that our method compared favorably to the alternative method, which is a method that specifically was also designed for multinomial data but was not factorial based. So similar observation applies if we consider small world type of network topology. But I said before that we can apply our method not only to multinomial data but to other data distributions, for example, to Poisson networks. So here we generate, again, observations where the underlying network was distributed according to Poisson, and our goal was to quantify, using the AUC, how does the inferred network match with the ground truth network. So here showed the baseline, which indicates the random prediction and a number of algorithms. So here the well-established, perhaps the most standard and very successful algorithms, which are Glassot, and Glassot is the algorithm which makes which is based on Gaussian assumption. And the log Glassot is similar to Glassot, which is a small transformation which pre-processes the data such that it relaxes the assumption of Gaussianity. And we can see here that two other algorithms which do more sophisticated pre-processing to transform data which is non-Gaussian to look like Gaussian data perform better than those methods that rely on Gaussian assumptions. And our FuseNet, which was designed, which in particular this case was used with Poisson model, performed the best. And this alternative method is another, which is also based for Poisson data, but is again not a factorial model behind it. So this was simulation study, and we were able to rigorously quantify what is the success of predicting links in the network. What we would also like is to evaluate the functional content of the networks from real data. So for that, we try to measure how well do functionally related sets of genes locate, or where do they locate in our inferred network. To do that, we overlaid the inferred network with the information with gene sets from gene ontology, and then we measure using various statistics how well do these functionally related sets cluster in the network. So two such measures, which are known from community detection, is flake-out degree fraction. So here, for example, this would be a network, and the the red highlighted part indicates the genes that belong to the apoptotic process in Go. So the flake-out degree fraction measures the fraction of genes within this set that have fewer links inside than to the outside. So for example, this gene has 5 neighbors, but 3 of them point outside to the cluster and just 2 point inside. So this is the one that counts. So there are 8 genes here in this region, and 3 of them have fewer links inside to the outside. So this would be the score for this particular Go term in the network. So an alternative is triangle participation ratio, which just indicates how many genes in the particular cluster here are members of a triad, so a triangle. So we can see here we have 7 genes in this yellow region, and 5 of them participate in triads. So this would be the score for this particular Go term in this network. So if I just show the results for these two particular measures, where higher score indicates more informative network in terms of its functional content, we can see that the networks that were inferred from fusion of different data sets performed better than the network that was separately inferred from individual data sets. So in particular here, the blue boxes indicate the network that was obtained from fusion of Poisson and multinomial data. So that means that we took the ICGC data and considered the mutation and RNA-seq data. And the performance here was substantially better than using either of the data set alone. And in terms of this alternative measure, we observed similar behavior. So what we can summarize from here is that in terms of functional content, fusion of somatic mutation and RNA-seq data from ICGC performed better than just considering networks that were inferred from a single data set. In terms performed better, what I showed is just two measures. What we also used is the recently proposed Santa tool, which measures the presence of how well or what's the strength of associations between Go terms and the inferred network. And so the fusion performs better than single data set if we employ the correct statistical model. By that I mean if we consider mutation data, then we should consider multinomial model, not Gaussian model. But even inference from a single data set is better than inference from a single data set that does not employ correct statistical model, or actually that ignores information about the underlying data distribution. Okay, so just a small part of the network that we obtained by fusing ICGC breast cancer data. So here is, for example, a transcriptional regulatory model that we were able to find. And GATA3 is an important regulator for breast cancer, which is associated with increased or reduced survival rate in the breast cancer patient. And another regulatory module is FLA1, and this one is important because we didn't find it when we inferred the network from any single data set, but it was found when we jointly considered both mutation and RNA-seq data. And one possible explanation for that would be that researchers were able to show that this module is associated with reduced survival rate when either one or more members of this module have changed expression levels or a mutation, which is exactly the two data sets that we considered. And the module was not found by considering any data set by itself. So to conclude, what we proposed is FuseNet, which is an off-the-shelf network inference approach that is able to fuse data from various potentially non-identical data distributions. It's based on the theory of Markov networks, which means that edges in the inferred networks indicate a type of dependence between genes that is stronger than Merck correlations that is a result of similarity-based approaches. And the high performance of the method is due to its two appealing properties. And the first one is that it can model any exponential family distribution, which means that it does not disregard the information about the underlying data distribution. And the second thing is that we achieve data fusion by sharing latent parameters between related data sets. Okay, so with that, I acknowledge my support to travel here, and you're also invited to the ECCB style poster that we have tomorrow. Thank you.
Thank you very much. My question is it possible to introduce multiple data sets in this model instead of two data sets that you...
Yes. So I just showed two data sets because it's easy to explain. Yeah. But in that sense, it's possible to have as many data sets as you have, as as you are able to tolerate with respect to computational time. This can follow from any combination of exponential family distribution. So you can have, let's say, you can have harnessing data in model it with Poisson model, then some mutation data with multinomial, but you can also have some categorical data or some Gaussian-based data sets.
Have you tested it on single cell data?
Not yet, but we would be very interested in doing that. Thank you.
Nice talk. It seems that you integrate several networks into union of the network. Have you tried the intersection of each network? I mean that have you tried any examination for the intersection of each network?
So we don't actually take the union of the network because all the networks, because we solve the optimization problem for all of them at the same time, because we have this coupling of latent parameters that are shared. But your idea of taking perhaps only the intersection would be interested, because perhaps this would be even more enriched core part. So it's a nice idea. Thank you.
I was wondering if there is an overlap between your network and the biological network. I mean, like KEGG or other source of network that are manually curated.
Okay, as I mentioned in the beginning, there are different ways of how we can construct network. So here, what is the interpretation of the edges in this network? The interpretation is the same as in undirected graphical models, which means that the two genes, let's say A and B, are connected if from behavior of genes B, we can get some information about behavior of gene A. By that, being more explicit means that the prediction model that is behind is able to use gene A to better predict the behavior of gene B. So in networks such as the gene interaction network, for example, from the Constanza paper 2000, 2010 are networks that are similarity based. So these networks measure what is, for example, the correlation between two genes, and then if correlation is above certain threshold, then two genes are connected. So other networks that you mentioned, such as KEGG pathways networks, can be manually curated networks, or the edges there have different interpretation than this here.
Last question.
Very interesting talk. Maybe something that so you mentioned it, but you didn't go into it. You say that you find these communities, so these modules. How exactly do you find those? So how do you pick them?
Okay, so I overlay the inferred network with the geo terms. Means that given the inferred network, communities or clusters are defined by the set of genes that belong to Go terms. So given that, we then quantify how community-like is this set of genes. So I showed here just two measures, which is this triangle participation ratio and flake-out degree fraction. And we use a couple more in the paper, and it was shown in the social analysis network research that these measures are complementary. So in addition to that, we also use this recently proposed Santa tool, which measures how strongly do Go terms cluster in the network that is the input to that is inferred by our method. So we evaluated the networks that are inferred by our method with respect to these community-like measures and the Santa tool.